Good morning.
Hello.
A warm and cordial greetings from the Oxford College of Science. Myself, Dr. Yogesh, heartily welcome all the participants to the day 2 of International Conference on Multidisciplinary Innovations in Science and Technology, MIST 2020. It is my privilege and honor to introduce the first speaker of the day, Dr. Anupam Paria, who has done his PhD in molecular biosciences from George August University, Germany, on the title Neurodegeneration Induced by Beta-synuclein in the context of neurotransmitter dopamine. His inclination towards research started in the year 2010 when he joined as an intern and later as research scientist at Biocon R&D, Bangalore, where he spearheaded experiments on upstream cell culture process, which went on to be approved by US FDA. His unbending acumen towards research propelled him further to work at Max Planck Institute for Biophysical Chemistry, Göttingen, Germany. He has various awards and honors in his ongoing research career and has a strong technical expertise in the field of cell culture, protein purification, structural biology, and bioprocess technology, to mention a few, and has a string of publications in high impact neurobiology journals. At this juncture, I would also like to proudly state that he has graduated from the Oxford College of Science, Bangalore, and went on to do his master's at Vellore Institute of Technology. Presently, he is a postdoctoral researcher at University of Texas, USA. My hearty welcome to Dr. Anupam Paria. I kindly request you to accept our invitation and take over the session and share your expertise with the participants. The session to you, sir.
Hello. Can everyone listen to me? Can you listen to me, Dr. Yogesh? Can you listen to me, Dr. Yogesh? Am I audible to everyone?
Okay. Yes, sir. You are audible.
Oh, great. Thank you so much, Dr. Yogesh, for a very kind introduction. I was, I'm quite glad that you explained everything very nicely. I'm very happy for that. And thank you so much. I would like to extend my thanks to Dr. Umesh, who invited me to this amazing international conference, the first conference where I would give a talk, and that too at the international level, and my country, India. And that's an amazing feeling. So, to begin with, I would just like to give you an introduction about myself. So, the, I basically belong from a place called Anantnag in Kashmir. So I was born in Delhi, and after traveling across India, like from Gujarat to West Bengal, I finally started my graduation in Bangalore. So I graduated from the Dayanand Sagar institutions, and Bangalore has been a wonderful place. Then I moved on to Vellore to do my master's in applied microbiology. And then I loved Bangalore so much, I went back to Bangalore and started working in Biocon in the R&D department. And as Dr. Yogesh mentioned very clearly, it's true that my inclination towards research spiked up when I got an industrial experience in Biocon. So, in the process of having my research interests getting spiked up and getting very much interested into research, I decided to pursue the career by doing my doctorate. So, I got a scholarship known as the AAD scholarship, which allowed me, uh, gave me a financial freedom to work as a doctoral student in a beautiful city of Göttingen. As you see here, this is the called as a Ganzliesel. So basically, every PhD student, like only PhD students who graduate, they have to climb up and kiss the Ganzliesel girl, and then they are known to be as doctorates. Not before that. There's a culture of the city, and now I'm in Dallas pursuing my postdoctorate studies. So, to begin with, this is the basic outline of my talk, where I would give you an introduction of neurodegeneration. Then I will introduce you to the research question which I'm going to answer in this presentation. I will discuss the results, the summary, and the overall outlook, how I see my research going, and then the conclusion. So, Parkinson's disease is like one of the second most common neurodegenerative disease of the central nervous system. The first comes the Alzheimer's, as everyone knows. The second is Parkinson's. And the major cardinal symptoms of PD are the resting tremor, where the hands and legs they shake unconsciously, the bradykinesia, the slow in movement, and muscle rigidity. So as you see on the figure here, as people can see my pointer, so this region which I've circled here and where my pointer moves is called, so basically this whole region is called as a midbrain, and this particular region is called substantia nigra, which is affected in Parkinson's disease. And the major hallmarks are basically the progressive loss of mostly dopaminergic neurons in this region, and the formation of like small leafy body like structures. So the leafy bodies are like circular shaped aggregates of protein and many other components like the plasma membranes, and also some of the fragments of the mitochondria and many other subcellular organelles. One of the components of leafy bodies is alpha synuclein. So the mutations in alpha synuclein is is actually known to cause Parkinson's disease, PD dementia, dementia with Lewy bodies, multiple system atrophy, and frontotemporal dementia. I know these diseases they sound very complicated. So it's just that these diseases are the neurodegenerative diseases, which lead to severe brain damage in due course of time. So it's a progressive neurodegeneration. Now, talking about the synuclein, the alpha synuclein as being the main component of living bodies. So this is quite abundant in the brain. It is mainly localized in the presynaptic nerve termini. So I assume that as people here are from different disciplines and backgrounds, so I assume that people have some knowledge about the neuronal cell. So it's just the endings of the neurons. This protein is mainly localized in this area. It's called as presynaptic nerve termini. And interestingly, this interesting small 14 kilodalton protein, it can aggregate to form oligomers, also ring-like oligomers, which further aggregates to form fibrils and amyloid fibrils later on, which then are found in the living bodies. Based on the literature and what people have reported is that these oligomeric structures they lead to several neuronal dysfunction, like the mitochondrial dysfunction and the dysfunction in the retrograde transport of proteins, the cellular trafficking, Golgi trafficking dysfunction, and this function finally leads to apoptosis. So this protein is like 14 kilodaltons. This can do wonders and can be one of the reasons where people suffer from Parkinson's disease. So this has three main domains: the N-terminal domain, the central MAC domain, and the C-terminal domain. So it's so based on the literature, what people report is that the central MAC domain is one of the main reasons why this particular protein, alpha synuclein, they aggregate into dimers and oligomers. And this N-terminal domain, there is a residue unit, a small motif, which is known to bind to the lipid membranes and might also play a role in aggregation. Another protein of the synuclein family is the beta synuclein. It is also like quite abundant in the brain, like alpha synuclein, and is distributed uniformly throughout the brain. It localizes in the same place as alpha synuclein and is known to play a role which inhibits alpha synuclein aggregation. This is beta synuclein structure where the this particular domain in the NAC domain is missing. So what people suggest is that because of this missing region over here in the NAC domain, the beta synuclein is less prone to aggregation. The last protein is the gamma synuclein, which also belongs to the synuclein family. So it is not involved in neurodegeneration as reported and is more prominently expressed in the peripheral nervous system. If you look at the structure here, usually N-terminal domain is mostly conserved in the synucleins, whereas in the gamma synuclein, the NAC domain is not completely present, and it lacks several regions of the C-terminal domain, and it's also much smaller than alpha synuclein or beta synuclein. So it's like the um, mainly dopaminergic neurons are degenerated, and alpha synuclein seems to play an important role. Therefore, this suggests a strong link between alpha synuclein and dopamine. Now, to give you a small overview of the metabolism taking place in a dopaminergic neuron, is that the tyrosine is hydroxylated to form L-DOPA. So this is L-DOPA. This is decarboxylated to form dopamine. Dopamine further can be packaged into small synaptic vesicles using the protein VMAT2. So this the red one is the VMAT2 which pumps dopamine into the vesicles. They can fuse to the membrane and the dopamine can be released into the synapse with the help of another protein called dopamine transporter. This cytosolic dopamine can actually be degraded by monoamine oxidase or ALDH into DOPAC, which can be further uh, converted to homovaleric acid using the enzyme methyltransferase. So this is the general mechanisms, the metabolic pathways which take place in the dopaminergic neurons in the context of dopamine. This dopac can actually be get oxidized to form dopaquinones, which is known to stabilize the synuclein oligomers, and it may cause the oxidation of methionine residues of alpha synuclein. So as a result of these, so this mechanisms why I am especially mentioning over here is because these oxidized dopaquinone species may result in the aggregation of alpha synuclein, which might lead to neurodegeneration. And also is known to be one of the causative reasons of neurodegeneration. So the cytosolic dopamine, when not sequestered by the vesicles, they can be degraded and they can be oxidized into dopamine quinone species, which further gets oxidized to form many other species like dopaminochrome from cytosolic dopamine. So now the beta synuclein. So the beta synuclein actually is reported that it can have an ability to aggregate in the dopaminergic neurons of the substantia nigra. And they can also cause neurodegeneration similar to alpha synuclein. So this suggests that there is or there might be a certain link between beta synuclein and dopamine, which has not been investigated so far. Therefore, the research question is dopamine or the dopamine metabolism linked to the neurotoxicity induced by beta synuclein? Now, to answer this question, the main aim is to establish a cell model with dopaminergic neurotransmitter phenotype and to study the neurotoxicity of beta synuclein in the context of dopamine. So these are like many neurons which are actually not dopaminergic. And my aim is to induce a dopaminergic neurotransmitter phenotype so that majority of them become dopaminergic, and then I can study the neurodegeneration induced by dopamine in, in these models in the context of beta synuclein. So I use the rat cortical neurons. So these cortical neurons are basically isolated from the embryonic uh, 18th day of rat embryonic pups. So the isolation is a bit complicated, but it is quite possible. So the majority of these cortical neurons are glutamatergic or GABAergic. So the idea is to transdifferentiate or reprogram the existing neurons into dopaminergic neurons. So this approach is called as lineage reprogramming or transdifferentiation. So these neurons are basically quite mature, like they get mature in 3 days. So we have to do a dissection of the rat pups. We have to isolate the cortex area of the rat pups and trypsinize them, separate the cells into single cells, and then plate them uh, on a well. So they are basically they stick to the bottom of the well. They are like adherent cell cultures. So they get mature in 3 days. So they start to mature in 3 days and get quite mature in like 8 to 10 days. So by maturation, I mean they are able to release and glutamate or GABA from these presynaptic termini, and they can actually talk to each other. So the majority of them are glutamatergic, and now I want majority of them to become dopaminergic. So the idea is to use an approach called transdifferentiation, which is the process where one mature cell can be reprogrammed into another mature cell. So there are a lot of transcription factors which play a role in patterning the dopaminergic fate in vivo. So the first one is like the LMX1A, which is quite central here. This further activates NUR1 or PTX3 to FOXA2 and through many other different transcription factors, which it together activate and induce the expression of tyrosine hydroxylase, which I refer here as TH, and it's the main determinant of a dopaminergic neuron. So here I use these transcription factors to introduce the expression of TH in these cortical neurons. So here is a Western blot where you see the endogenous levels of NUR1, ACL1, LMX1A, PTX3 at different time points. So it's surprising to note that the transcription factor over here, PTX3, is quite present endogenously. So these transcription factors are mainly present in the dopaminergic neuron, as PTX3 is constantly present over here, but still they are not dopaminergic neurons. So which could be that the NUR1, ACL1, or LMX1A levels are different. So I used adeno-associated viral vectors to introduce these transcription factors in the cortical neurons, and then analyze the expression of tyrosine hydroxylase using immunocytochemistry at these different time points. So interestingly, I found that the transcription factors ACL1, NUR1, and LMX1A induce the expression of TH in these cortical neurons. So, so these are the TH positive neurons over here after 2 weeks. So not only they induce the expression of TH, but also other dopaminergic markers like aromatic amino acid decarboxylase, vesicular monoamine transferase 2, and dopamine transporter as well. So this was very interesting and remarkable result, which really got us excited. That yeah, I mean these neurons can actually be reprogrammed to dopaminergic neurons, which was never done before. So now I analyze the numbers of these TH positive neurons, and I found that although it's really possible to reprogram these cortical neurons towards dopaminergic neuron, uh, TH positive neuron, the numbers are quite low. So I get around like 10 to 15% of TH positive neurons after 3 weeks or after 2 weeks. And uh, so these numbers seem to be a bit low for the study. However, This was a very interesting finding, so I further went ahead and I found that just as because only 10 to 15% of the population were Th positive, suggesting that only a subpopulation of these cortical neurons have become dopaminergic. So the question is next, which type of cortical neurons basically transdifferentiate to dopaminergic neurons? Because at the majority of them are glutamatergic, only 15% can become Th positive. So I was very curious to know which type of cortical neurons reprogram. I have found that these the TH signal basically co-localizes with the marker GAD65, is glutamate decarboxylase. It's basically one of the markers of a GABAergic neuron. So as they co-localize, this was very clear that the GABAergic neurons basically transdifferentiate towards dopaminergic neurotransmitter fate. And I also found that the glutamatergic neurons, which are detected by the marker CAM K2 beta, they do not co-localize with the TH, suggesting that the glutamatergic cortical neurons do not transdifferentiate. So I just counted the numbers, and this suggests that basically, as the TH number of TH positive neurons they increase, the number of GABAergic neurons decrease, which means that the H positive neurons are originated from GABAergic neurons. So, so it's it was very clear that the transcription factors ACL1 or 1 LMX1A they are toxic to specifically glutamatergic neurons, as the uh, number at day in vitro 7, the number of GABAergic neurons that remain the same. And these GABAergic neurons are reprogrammed to Th positive dopaminergic neurons. So these are the dopaminergic neurons which increase by day in vitro 14 and are almost the same by day in vitro 21. So to summarize the first part, the transcription factors ACL1 and NUR1 were sufficient to induce the expression of Th in just 10 to 15% of the cortical neurons, which are mainly GABAergic. And they also induce the expression of ADC, VMAT2, and DAT. So the transcription factors ACL1, NUR1 overexpression, they are actually toxic to the glutamatergic neurons only. So this study basically uh, proves the concept that the neuronal transdifferentiation of postmitotic neuronal cells can be achieved. Now, as I mentioned before, that only 10 to 15% of these neurons became Th positive. So this model was not uh, great enough to study the in neurotoxicity induced beta cell nuclei in the context of dopamine. Therefore, another approach was employed where the dopaminergic neurotransmitter phenotype was introduced in these cortical neurons. So the L-DOPA, as I mentioned before, while explaining the metabolism, the L-DOPA, AADC, and VMAT2, they are the core dopamine producing machinery. So as dopamine that produced is sequestered into vesicles and released by dopamine transporter and also uptaken by dopamine transporter back into the cells. So the design of the experiment was that I would introduce these AADC and VMAT2 using viral vectors, the adeno-associated viral vectors at the in vitro 0, and also DAT and VMAT2 over here, and apply L-DOPA as a substrate or dopamine to increase the levels of dopamine cytosolically. So, uh, so the first model where ADC and VMAT2 were introduced and L-DOPA was used as a substrate, I see that there was a huge increase in the intracellular levels of dopamine. And also, the dopamine was metabolized into TOPAC and HVA, which was detected intracellularly and also in the supernatant. So now, when dopamine was used as a substrate in the medium in the cells expressing dopamine transporter and VMAT2, the dopamine was readily taken up and sequestered into the cells with the help of dopamine transporter, which spiked up the intercellular levels of dopamine over here. And the supernatants, the levels of the supernatant or the levels of all the metabolites and dopamine in the supernatant were also detected. So it appeared that the system and the model worked quite good. Therefore, I used the first cell-based model and I introduced alpha synuclein, beta synuclein, gamma synuclein, and EGFP. So EGFP is and gamma synuclein are the controls. So as people might be, some people might not be aware that EGFP is basically a green fluorescent protein, and this is an enhanced version of EGFP, which is not toxic to these neurons. So and then cell counts were taken at these time points. So I found that dopamine actually enhanced the neurotoxic potential of alpha and beta synuclein, whereas the controls were good, as AGFP and gamma synuclein were not toxic. The white bars are basically the bars where dopamine was not produced by the cells, and the black bars are the bar which represent that cells producing dopamine. So this suggests that these two bars uh, uh, at day in vitro 19 suggest that the dopamine production it enhances the neurotoxicity of alpha and beta synuclein. Now, using the second cell model, when the cell counts were taken, the controls were good, where there is no such significant difference between the white and the black bar. Again, the white bar are the cells where dopamine was not taken up, whereas the black bars are the cells where dopamine was taken up. So again, at the in vitro 19, when the dopamine was taken up, again, this suggests that dopamine, when it takes up, dopamine enhances the neurotoxic potential of alpha and beta synuclein. So now uh, I went on ahead and with collaborating with one of my group members, Christian Leite, we performed and studied the endogenous network activity. So the what is network activity? It's basically like how neurons they talk to each other. Like when they talk to each other, they fire. So it's like they release neurotransmitters and they take up. They release and they take up. So the frequency is decided by a certain frequency in a brain. But when alpha synuclein is expressed more at toxic levels, the frequency changes. So we detect the change in the frequency in this cell model. So the question here is that does dopamine production affect the endogenous neuronal network activity in the presence of alpha and beta synuclein? So the design is that we use some calcium sensors. So the so as the neuron is depolarized or as it fires, there is a calcium influx which is detected by these calcium sensors. So each detection is a single burst, and many detections are result into many bursts. which we detect. So again, in this cell model, we introduced a calcium sensor, which is also called as GCAMP. We introduced synucleins, and we analyzed the number of pulses per minute at these time points. So based on the results, I found that dopamine production or uptake did not affect the endogenous network activity of these neurons when alpha and beta synuclein are expressed. So now I was quite interested that as dopamine enhances the neurotoxicity of synucleins, how should I study this at a molecular level? So, um so I decided that I think NMR would be a very good tool. So basically, just to put in a easy way, the idea is to study the interaction of dopamine with synuclein. So if this is an amino acid residue, and if a ligand comes in, uh, interacts with this residue, there's a shift of this peak to here, which is represented as chemical shift perturbations over here. So this is a shift of the chemical peak in the presence of a ligand which is detected. Therefore, the question here is, does dopamine interact with alpha and beta synuclein? So the NMR studies reveal that yes, dopamine does interact with alpha and beta synuclein, but dopamine here in alpha synuclein, it interacts all majority of the interaction with alpha synuclein is in the C termini, whereas with beta synuclein is all over the place. So so with collaboration from Professor Griesinger from the Max Planck, uh, we performed these studies and we calculated KD. So KD is basically the ability of a ligand to get dissociated from a molecule. So higher the KD, lower is the affinity. So based on the KD values which you see over here, it's very clear that the binding affinity of dopamine is much stronger with alpha synuclein than beta synuclein. So this suggests that could be that the, the structural differences in alpha and beta synuclein could be responsible for the different spectra from the NMR and the different binding affinities. So it is reported in the literature that in alpha synuclein, YEMPS is one of the regions where dopamine interacts, and in beta synuclein, it's not exactly YEMPS. It's different. So could be this is one of the reasons why might be the dopamine it as it interacts all over, it is not responsible. It does not it is does not bind very strongly to this region. It is just all over. And also the glutamic acid over here in the NAC region is absent in beta synuclein, as this is one of the amino acids which is known to play a role in the aggregation of alpha synuclein. Now, to overall summarize, in the first part, we saw that the neuronal transdifferentiation of mainly the GABAergic cortical neurons, which are terminally differentiated, can be achieved by the transcription factors ACL1 and NUR1. So the dopaminergic cell model was developed. It was quite found to be quite robust, which has good intracellular levels and can release dopamine into supernatant. It was developed by mimicking the dopaminergic neurotransmitter phenotype in the cortical neurons. Interestingly, in this cell model, it was found that dopamine enhanced the neurotoxic potential of alpha and beta synuclein. So, yes, the binding affinity of dopamine was not much stronger to beta synuclein than it was for alpha synuclein, which suggests that maybe the metabolites of dopamine play an important role in the interaction with beta synuclein and inducing neurodegeneration. So, now the next question is, why does dopamine enhance the neurotoxic potential of beta synuclein? So, can dopal, which is one of the metabolites of the dopamine pathway, can this be one of the reasons? Now dopal is very unstable as it's an aldehyde, and it is involved in the dopamine degradation pathway. It is known that um, dopal can actually interact and covalently modify the lysine residues present on the alpha synuclein and result in alpha synuclein oligomerization, which might lead to neurodegeneration. And these lysine residues are also found on beta synuclein at these points. So, could this be one of the reasons that can dopal interact with beta synuclein and result in the oligomerization of beta synuclein and thus lead to neurodegeneration? This is an interesting question which needs to be answered. Now, next interesting role is would be of reactive oxygen species. So, can alpha synuclein, which in the presence of reactive oxygen species, they increase the thiol oxidation in the mitochondria and results in the cytochrome C release in the cytoplasm. So, also the cytosolic dopamine can further be degraded into dopac and dopaquinone, which which might release the hydrogen peroxide and be involved in the reactive oxygen species. Therefore, this mechanism can be very well valid for beta synuclein as well. However, this needs to be tested and studied further. So, therefore, going back to my this beautiful image of a brain, I conclude that the results in this study it provides new perspectives, like in the neurodegeneration in Parkinson's disease, and I strongly the result they strongly suggest that not just alpha synuclein but even beta synuclein should be considered an important entity in Parkinson's disease and its associated neurodegenerative disorders. So, with this, I would like to thank first my supervisor. I have two supervisors. First one is Sebastian, who directly supervises me, and I belong to the lab of Professor Bear. So I would like to thank Sebastian for his excellent support throughout my PhD. I would like to thank Monica, Sonia, Claudia, and Elisa for their excellent technical assistance all throughout, without which it would have been impossible to do many experiments. And these are my lab members uh, who have played an important role in the progress of my PhD. They helped in the discussion and also played an important role as co-authors and performed different experiments which led to the publication. I would like to thank Professor Bear for giving me an opportunity back in 2015 for working as a PhD student in his laboratory. I would like to extend my thanks to Professor Browse and Professor Otiero for, for being a part of my PhD project and an extended PhD committee where their inputs really helped me a lot. I would like to extend my thanks to Professor Griesinger, to Dr. Kalyan and Dr. Stephen Becker, who actually they are the main people for the NMR experiments who are very much specialized in NMR. Professor Griesinger gave me an opportunity to work on this project in his department, and with the help and a lot of assistance from Dr. Kalyan, this was a success. Finally, I would like to thank Dr. Umesh. So Umesh sir has been with me. So uh, he knew me for a very long time since 2005, as he mm, was one of the main motivators of me of biochemistry basically. So Umesh sir really helped me, and not just me, many other students. And I was the lucky one where I really picked up his teachings quite well and have excelled afterwards. So I would like to thank Umesh sir for his support and during my graduation, for his teachings, and a really nice time, and especially for inviting me to this amazing international conference. I would also like to thank the organizing committee of the Oxford College of Science, where I think they have played a very important role. It's so difficult to organize an e-conference. I applaud to the organizing committee, to the board members, to everyone who played a very important role in making this conference a success. Thank you so much.
Thank you, Dr. Anupam Paria, for your wishes. There are few questions posted in the
Yeah,
chat box.
in the chat
I
box.
wish you will want to answer them.
Let's see.
This is a question from Dr. Umesh.
Oh, yes.
How synucleins aggregation induced by dopamine? Is it age factor or any other specific reasons for it?
Uh, okay. So, yeah, that's a nice question. So, um, so, there are a lot of factors which are reported to be responsible for aggregation of synucleins. Dopamine is one of them, and the the metabolism of dopamine produces a lot of reactive oxygen species, a lot of stress to the cells, uh, and it is basically known to interact with the glutamic acid in the NAC domain of alpha synuclein, and also in the YEMPS region of the C termini. So, as a result of the interaction, these alpha synuclein, which are basically soluble, so after dopamine get they bind the dopamine binds to alpha synuclein, they like stick to each other. These monomeric alpha synuclein stick to each other, and they oligomerize and form insoluble fibrils, which accumulate into Levy bodies. So this is just one of the hypotheses, and has a lot of work has been done and proved that this is really true in the context of dopamine. With age, yes, with age, there are a lot of toxins which are not washed away, and there are the protein degradation pathways which deteriorate, the efficiency of which deteriorates with age. So as the protein degradation pathway do not work so good, so like the ubiquitin proteasome system or the lysosome or the autophagy lysosomal system, if they do not work so good, these alpha synuclein which tend to aggregate, they are not cleared up from the cells, as a result of which they continue aggregating and forming clumps of fibrils inside the cells. And it has been reported that these might act as a prion like species, which might spread from one cell to another cell. And therefore, they might spread to the whole brain, which is very, very dangerous, and work has been going on with them. I see another question. Is it, uh, did my answer help?
Yeah, yeah.
Okay. So there's another question that are any specific reasons why some neuron didn't reprogram? Yeah, that's a very excellent question. Um, the the answer to this is that I do not know at the moment. Why? other neurons did not reprogram? I have no answer to that. So, and I only have an assumption. Is the assumption is that uh, the, uh, the 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 cortical neurons, like the glutamatergic neurons. So it could be that there are so the cortical neurons are different from GABAergic neurons. So there could be many regions in the heterochromatin which are not accessible to ASCL1. Is one of the transcription factors which actually is known to access the uh, the promoter region of the DNA so that other transcription factors can bind. So because of tightly packed heterochromatin, it could be possible that this transcription factor could not access the promoter of dopaminergic markers, as a result of which they do not become dopaminergic. But I do not have an experimental proof for that, so this is just might be an assumption. A lot of work has to be done on that.
Thank you.
Mm-hmm.
I
Yeah,
think there are
thank
no
you.
more questions.
Okay, great.
I thank
Thank
Dr.
you.
Anupam Paria for a scintillating talk on understanding Parkinson's disease at molecular level. Sir, you are
Thank
an
you.
excellent orator, and I can confidentially say that the participants would have got immensely benefited from your talk.
Hope so.
I sincerely wish you success in your future research in this arena, leading to positive breakthroughs in this field.
Thank you. And I would like to also mention one
Paria.
more thing. I would also like to mention one more thing to the students. If there are students listening, is that please feel free to approach me if you need any help with career related or how to progress. Should you go to an industry, research, how to do things. So I can also share my experiences if approached by students. So I have my email ID on the abstract. Please feel free to approach me. I'll be happy to
Surely,
help.
surely. Thank you once again.
Okay. Thank you so much.
Participants are requested to stay back in the meeting for oral presentations by research colleagues.
Good morning, everyone. I, Dr. Gopal Reddy, welcome you all to the oral presentation session 4.
My poster. As we know, hello everyone. I'm glad to present this work before you. First, I would like to thank MIST for giving me this opportunity to showcase my work. Alright, today I'm going to present my project innovation called EDIT. EDIT stands for Enhanced Device for Identifying Intrusion and Tag for Humans. First, I would like to explain edit block diagram. Block diagram consists machine fitted with edit device. Edit consists of microcontroller unit with Doppler sensor module and ESP room 32 module and variable device called X-band which communicating with microcontroller. By Bluetooth BLE technology, the data received from the Doppler and X-band is processed by microcontroller. The data sent to cloud or server database and then to dedicate a dashboard. Now edit for identification of human intrusion by Doppler sensor fitted to machine with the controller. The signal is identified by controller, then enables Bluetooth read and scan for nearby Bluetooth devices. This Bluetooth device detect within the range based on unique ID provided for similar Bluetooth bands. One which is very nearer to controller is taken and thus the intruder with the machine is identified. The signal read by Bluetooth receiver are further classified based on the signal strength. One which is nearer will have a Bluetooth signal transmitted with greater signal strength identified in decibels, hence identifying the human in access to machine. These edit devices are connected and configured for an array of machines. Machines are thus identified and various data like machine operation, worker login, logout, efficiency, yield of machine operated by each individual worker can be tracked. Edit is classified in such a way that every edit is operated in master mode and Bluetooth expander made to work in a slave mode. And it is dependent and edit is operated or read is thus independent. This and the Doppler sensor, its range and area covered are configured carefully. All the possible data above mentioned are captured on real-time basis and sent to the database or cloud by the ESP32 controller and the data logged or read or monitored. With the ESP32 controller is connected to Wi-Fi so as to send the data to the database or cloud. And the working of the Doppler sensor geometrical representation you're able to see here with the Bluetooth range and the representation in a geometrical which we are using in edit Doppler sensor. This Doppler sensor is uh, manufactured by Japanese. That port number is NGR4265R is a 24 gigahertz. It is a unlicensed ISM band. The microwave intelligent motion sensor for short distance low speed applications. Specifications of this Doppler sensor is radio frequency is 24.5 to 24.25 gigahertz and the maximum detection distance is 10 meters. Antenna angle is 70 degrees or 90 degrees and detectable speed range is 0.25 to meter per second. Detection output method is UART communication. For this Doppler sensor, input voltage is 3 to 5.25 volts and current is up to 60 milliamps. This is the circuit diagram of Doppler sensor. It is communicating with the microcontroller with the UART communication. This is having dedication LEDs for approaching and leaving detection and also it contains system power indication. And the X band is a wearable smart electronic band which is configured to connect edit with an unique identification ID provided. X band uses Bluetooth low energy protocol to connect edit. This block diagram of X band is contains USB micro and charge controller IC and the rechargeable battery and low dropout is used to power STM32 Bluetooth SoC and there is a dedicated Bluetooth enable button and the LC. Uses of this edit is The proposed concept of EDIT is mainly focused for industrial sector problem. Further development in a later stages, the complete setup can be reconfigured and can address various real-time problems related to human identification and sensing remotely, maintenance of machines, automation, production yield and efficiencies are estimated. The EDIT is a inclusive application of IoT and embedded sensor networks. This efficient design and can be operated in variety of environments. First of a kind device that approach for solutions to various industrial sectors, production related and maintenance problems and more and more. Thank you.
Hello everyone, I am Basant Kumar from Oxford College of Science. I am going to present a study on evaluation of cytotoxicity and apoptotic potential of beta-valgaris root extract against SCT-116 cell line. Background of the study: cancer is a group of diseases which involves uncontrolled proliferation of cells. Cancer is due to accumulation of gene mutation which regulates cell growth and division. It is a group of more than 100 diseases that develop across time. Cancer is the second leading cause of death globally, accounting for an estimated 9.6 million deaths or 1 in 6 deaths. Colorectal cancer is the third most commonly occurring cancer in man and the second most commonly occurring cancer in women. Plant-derived drugs are designed for anti-cancer treatment as they are natural and readily available. Vita vulgaris is a plant which belongs to family Amaranthaceae. Beet have been used in folk medicines all over the world for different reasons. A lot of research has indicated that increasing consumption of plant foods like beetroot reduce the risk of obesity, diabetes mellitus, and cardiovascular diseases. The beetroot plant demonstrates benefits for cancer treatment. Hypothesis of the study: the aqueous extract of beetroot contains valuable and active compounds such as carotenoids, flavonoids, phenols, and alkaloids. In the present work, the cytotoxic effect of aqueous beetroot extract studied by MTT assay against human colon cancer cell line, that is SCT-116, showed significant cytotoxic activity. Cell death analysis and cell cycle study was conducted using flow cytometry. The data obtained in the study revealed the therapeutic value of beta vulgaris and the scope for further development of cancer drugs. Objectives of the study: first, to study the anti-cancer activity, that is apoptotic activity of beta vulgaris on SCT-116 colon cancer cell line through cytotoxic assay. Second, to determine the cell analysis on SCT-116 cell line by flow cytometry. Materials and methods: Beta vulgaris is a member of the amaranth and goosewood family. Beetroot is used in the ancient Rome and in the herbal treatment of cancer. It contains high concentration of red betalains, vitamin C, tyrosine, iron, and folic acid. SCT-116 cell line. SCT-116 cell line is a human colorectal carcinoma cell line initiated from an adult male. The cells are adherent with an epithelial morphology. Steps for the study: preparation of crude extract. 500 grams of dried powdered plant samples were extracted using water as a solvent. Phytochemical screening. The presence of the functional groups in the extract were analyzed through biochemical methods. Cell culture studies. Toxicity of the compounds was verified by performing by MTT assay. Apoptosis study to analyze the mechanism of the cell
இப்போ அடிக்க வைக்க முடியாது. அம்மா கொஞ்சம் படிக்கணும்.
cycle study
ஏதாவது ஒண்ணு
to
பண்ணணும்னா
analyze
கரெக்டா
the
பண்ணணும்.
cell cycle.
சரி, ஓகே.
Results and discussion. First, phytochemical screening. Through phytochemical screening, it was found that flavonoids and glycosides were present in the beetroot extract.
அத பத்தி எல்லாம் ஆராய்ச்சி பண்றாங்க.
Healthy cells in human colon cells. Fourth, cell cycle study. The cells treated with standard, control, and beta vulgaris extract with the concentration of 25 micromolar and 200.59 mg per ml respectively are showing high percentage of cells at S phase and J2M phase. Hence, the cell cycle got arrested at J2M and S stages. Vita vulgaris extract exhibiting prominent cell cycle phase arrest similar to the standard control camptothecin on SCT-116 cells. Conclusions of the study: the observations strongly suggesting us that the test compound namely beta vulgaris extract, may have possible therapeutic potential against human colon cancer cells based on the dosage of the drug after the incubation period of 24 hours, and further studies need to be performed to evaluate the molecular mechanism of action behind the anti-cancer potential of both test compound against the colon cancer cells in vitro conditions. The observation suggests that the test compound beta vulgaris extract induces the significant necrosis and apoptosis compared to healthy cells and colon cancer cells. The extract beta vulgaris may have therapeutic potential against human colon cancers, and further preclinical studies have to be done to confirm the mechanism of action on SCT-116 cells. The cells treated with standard control and beta vulgaris extract with the concentrations of 25 micromolar and 200.59 microgram per ml respectively are showing high percentage of cells at S phase and G2M phases. Hence, the cell cycle got arrested at G2M and S stages. Beta vulgaris extract exhibiting prominent cell cycle phase arrest similar to the standard control camptothecin on SCT-116 cells. These are some of the references I used. Thank you.
Good morning, one and all. I am Shweta, pursuing second MSc Bioinformatics from Sri Krishna Arts and Science College. My paper is on pathway reconstruction of Pseudomonas aeruginosa by the integrated analysis of genomics and metabolomics, which is an initiation towards identification of therapeutic markers. Since we have selected the organism Pseudomonas aeruginosa, let's have a glance through this organism and let's see what is the property that we have selected for its study. So, this organism is an encapsulated gram-negative bacterium that can cause disease in plants and in animals, including humans. One of its unique property is that it is a multi-drug resistant pathogen and shows advanced antibiotic resistance mechanism. So this is the property that we have selected for the study, that it can cause disease in plants and animals. At the same time, it is resistant against multi-drugs and antibiotics. So we thought that how we can prevent this organism from infecting the host cells. Apart from that, the other factors like it thrives not just in normal atmosphere but also in low oxygen atmosphere. Therefore, it can easily infect damaged tissues. Above all, it is also found on moist surfaces. Therefore, it is found on and in medical equipments, causing cross infections in hospitals and clinics. Therefore, it is found in wide areas. Thus, we have concluded in studying this organism Pseudomonas aeruginosa. So, for the purpose of studying the Pseudomonas aeruginosa, we need to study the mechanism of this organism within the host cell. For this purpose, we have gone for studying, or we have opted the method of pathway reconstruction or network reconstruction. So, this is the method that allows an insight depth into the molecular mechanism of a particular organism, and we can find out the metabolomic pathways. And actually in reconstruction, it actually breaks down the metabolomic pathways into their respective reactions and enzymes, and then we can analyze within the perspective of the entire network, whereby we'll get the metabolites and enzymes, their forward and backward reactions, as well as the free energy change. All this information could be obtained from this network reconstruction or pathway reconstruction. So for this purpose, we have used wide varieties of bioinformatics tools like David, CAS and WebGestalt. So this actually is a resource. It is a database for annotation, visualization and integrated discovery. As I said, it is a resource which consists of wide varieties of tools and that provides functional interpretation of large list of genes derived from genomic studies like microarray and proteomic studies. So the different tools are functional classification tool, functional annotation tool, gene ID conversion tool, gene name viewer and pathogen genome browser. So these are the different tools that is available in this David tool, and with that we will get a functional information regarding this gene. Then comes the next tool, namely CAS. It is CAS means KEGG automatic annotation server, and it also provides functional annotation of genes by BLAS, where comparison is done against the manually curated KEGG gene database, and also the result will contain KEGG orthology assignments, and also it will contain automatically generated KEGG pathways, and that is our area of interest. We require the pathways and its reconstruction is what we require. So on the basis of that, we have tabulated the results containing the metabolites as well as the enzyme, their free energy change, everything has been found out. Also we have done a graphical representation of this metabolites and enzyme on the basis of the free energy change. So from this, what actually we concluded is that specific metabolites like glucosyltransferase and oligosaccharides, their association is found to be an active cascade that could drive the metabolism of Pseudomonas aeruginosa. This is what we have inferred from the all the tabulations we have done, all the results that we have obtained. So the association of these two metabolites and enzyme, that is glucosyltransferase and oligosaccharides, their forward reaction will result in an active cascade to drive the metabolism of Pseudomonas aeruginosa. So if we are able to find out certain inhibitors that could inhibit the forward reaction of this particular metabolite enzyme association, we can prevent the Pseudomonas aeruginosa from infecting the host cells. So this is the idea that we have found out, or this is what we have concluded from the pathway reconstruction of this organism. Thank you.
Namaste and good day to all. The title of the paper is Optimization of the Thermostable Alkaline and Acidophilic Proteases from Bacillus species using various ways. Myself, Preeti S. Kumar Mat, Assistant Professor, Department of Biotechnology, Basaveshwar Engineering College, Bagalkot, and the project associates are Harsha Raghuram, Veeranna, Malkan Gowda, and Chetan. Introduction of the paper. Here, the enzymes are highly specific catalysts which have many industrial applications. Proteases occur in all organisms from prokaryotes to eukaryotes and to the viruses. Proteases are the main enzymes produced from microbial sources, of which only few are recommended as commercial producers. In view of their wide applications in various industries, protease enzymes occupy an important position. Alkaline proteases constitute 60 to 65% of the global industrial market. These proteases are the single class of enzymes widely used in detergents, pharmaceuticals, leather, food, and agriculture industries. Bacillus species are the main producers of extracellular proteases, and industrial sectors frequently use Bacillus species for the production of various enzymes. The main aim of the present study was to isolate, purify, and characterize alkaline proteases from Bacillus species from waste products and optimize the conditions for maximum production of extracellular proteases. The materials for the work is casein agar of 2%. It is nutrient agar with the required amount of casein. Next is a soil sample which was collected from the rice field. Dairy and market waste. Dairy effluent is collected from the dairy and the vegetable waste is taken from the city market, Bagalkot. Methods to optimization of the thermostable alkaline protease production. The steps of experiment goes with the collection of the sample, screening of microorganisms, culture and growth conditions, protease assay through Lorentz method. Purification of the protease obtained is done by HPLC and page. First, the sample collected from various sources. Here, sample 1 is dairy waste from dairy. Sample 2, vegetable waste from the city market. Sample 3, soil from paddy field. Screening of microorganisms. Sample 1 is dairy waste. For isolation, dairy waste was collected from the dairy. 0.2 ml of the sample was spread on casein agar plate and incubated at 30 degrees centigrade for 7 days. Enriched sample was plated over nutrient agar containing 0.4% gelatin. After incubation for 24 hours, plates were flooded with 1% tannic acid. Colonies showing clear zone were picked and purified, and isolates were screened for protease production by using casein digestion method. Vegetable waste. The vegetable waste were collected from city market. Collected waste were washed with sterile distilled water and cut into small pieces. These were taken in a china bowl and crushed along with water, and the juice was extracted. 0.2 ml of the juice of the sample was incubated on casein agar plate. The inoculated plates were taken and incubated at 37 degrees centigrade for 48 hours. Observe for the zone of clearance, which indicate proteolytic activity. Based on the morphology, the isolates were identified as bacillus species. The bacterial cultures were purified and maintained on nutrient agar slants having 35 degree centigrade. Sample 3 here is the soil from paddy field. Soil samples were collected from the paddy field, which is sun dried. 1 gram of the sample was suspended in 100 ml sterile distilled water, agitated for 45 minutes on a shaker at 50 degrees centigrade. Here, the 0.2 ml of the sample was spread on a casein agar plate and incubated for 30 degree for 7 days. Same here, the procedure goes wherein it was spread over the agar plate and flooded with 1% tannic acid, and the zone of clearance were observed, and this was picked and purified, and isolates were screened for protease production by using casein digestion. Protease which was being isolated was taken for the assay was done by Loddy's method. 1 unit of protease activity is defined as the amount of micromoles of tyrosine equivalent released from casein per minute under experimental conditions. Optimization of the culture conditions for production of enzyme with effect of pH, effect of incubation temperature, effect of incubation time, agitation speed, that is for 50 to 250 rpm. Results and discussion. The parameters that largely have impact on enzyme production are incubated, impulse speed, pH and incubation temperature, and nutrient supplements. In the present study, an attempt was made to optimize the production parameters for a bacterial isolates from paddy. Here comes the result. Effect of pH on activity of proteases. For pH stability, the enzyme was pre-incubated at various pH values like 2, 3, 4, 5, 6, 7, 8, 9, 10 and 11 overnight and determined the activity and found that pH 10 as the optimum pH value that showed maximum activity. So here 0.54 is the activity which is observed at pH 10. Growth of colonies at different temperature conditions. The optimum temperature determined was 45 degrees centigrade at which maximum growth was observed. So this is 45 degree. Maximum number of colonies were found to be 72. Effect of incubation time on protease. Optimum incubation time period was 160 hours per day, that is 7 days for maximum yield. So it was 7 days 168. The maximum yield was measured at 7th day, that is 168 hours. Effect of agitation on protease. So agitation speed was done at 50 to 250 rpm was studied, of which 200 rpm found to be the suitable speed for growth of colonies. So here is at 200 rpm the maximum number of colonies were 36. Application of proteases. In medicine, proteases are used for the treatment of blood clots in the ischemic stroke and wound debridements. Pharmacology and drug manufacture. It is anti-protozoal and anti-cancer activity. It's also used in the laundry and dishwashing detergents. Used as hard surface cleaning formulations. Also used for the contact lens cleaning formulation. Used in waste treatment. Industrial applications are many. Used for the fermentations. It is also a chondroitin and heparin production field. It's also used in the animal feed additives. Proteases play a very important role in digestive supplements, improves digestibility, solubility, flavors, palatability, and also improves many processes of life. Coming to the conclusion, we can use enzymes such as protease in a purified form in different industries such as textile industry, leather, detergent and other food processing industries, which will promise to be a cheapest source. Utilization of waste will minimize the deposition of waste matter in the environment, so as the chemical catalyst usage will be reduced, and on the other hand it is economically beneficial. The neutral protease produced during log phase will play an important role in food industries for making cheese. Of all proteases, alkaline proteases produced by bacillus species are of great importance for detergent industry due to their high thermostable and pH stability. These are some of the references that have helped to prepare the topic, and also the work was done using many of the research papers. Thank you for the opportunity. Thank you one and all.
Sir, myself Kunal Bera, a B.Pharm student of the NIT University, West Bengal. Here I am presenting a review on the pharmacological activity of Sosota obulata, which is belonging to the Asteraceae family. It is also called the spiritual queen of the night. As we all know, the medicinal plants are indeed a well-tested medicine, modern medicine, nutraceuticals, food supplement, Vedic medicine, folk medicine, pharmaceutical intermediates, and the chemical entities. Drugs. The herbal drugs are used in the traditional Indian system of the medicine and also in the other system of the medicine in the world for the time immemorial due to low cost, the effectiveness and no or low side effects. These attributes of the so-called the natural medicines are the attracting the attention of the increasingly large number of the people of the world. The World Health Organization, also called the WHO, reports that the 80% of the population in the developing countries are still dependent on the traditional and the folk system of the medicine. 85% traditional medicines are prepared by the use of the plant extracts, and that nearly 70% of the prescribed human medicines are derived from the plant. In that slide, you can see the taxonomical classification as the kingdom, division, class, order, family, genus, species is also. And the Sorceria oblata is also called the Brahma Kamal. It is a perennial plant with the stem, the 15 to 45 centimeter long leaves are belong to blunt lanceolate in shape, and the leaf margin are the toothed. The lower part of the leaf is stalked, and the upper part is the half clasping with the blade. The expanded flattened part 1.5 to 2.5 centimeter long. The densely packed the cluster of lower or flat and is supplemented within the volucral bracts and with the black margin and the tips, and the entire flower head is covered by a large pale yellow or boat-shaped and peppery bracts. Geographical distribution, you can see the geographical distribution of this plant is limited to the high mountain, the ranges of the Kashmir, Himachal Pradesh, Garhwal, the Chamoli, Himkut, the Sikkim in India, and in the Pakistan, Nepal, Bhutan, Tibet, and China. Snow lotus is restricted to the high alpine reaches up to 51,000 meters. The Brahmokamal mainly grows in the alpine midwest, the glacier slopes along the side of the lake, and the streams, the alpine screes, the rocky slopes, and some other high mountain, the habitat the preferably alpine, the meadows, the screes, and the dry rocky slopes. It is nearly endemic to the Sikkim Himalayas. Other than the India, it is the very sparsely distributed in the Nepal, Bhutan, and the Tibet. So the medicinal plant have been traditionally used to treat the humans in the various health problems since the ancient period. And the systemic synthetic drugs, the Sorceria oblata is one of the important medicine plant which is being used from the ancient time to till that for the treating different disorder. That it is commonly known as the Brahmokamal, which belongs to the family the Asteraceae. It is mainly found in the tropical and subtropical region of the world. Our main aim, these articles aim to review a pharmacological activity of the Sorceria oblata based on the extensive literature survey. And you can see the several vitamins and mineral are found in that flower, like the vitamin A, C, vitamin B2, B1, and magnesium, ferrous, etc. And in the method part, you can see up to date till June 2020, the search was done is in Google Scholar, the Scopus, the Web of Science, the Springer link, the ResearchGate publication, the prompt, the science at the direct database with the several keywords to select all the possible and the informative. Then the evidence to received in this paper, and the search yielded 120 articles, and after exclusion a total of the 57 were finally selected to perform the study. In a result part, you can see the Sorceria oblata is the important source of the many active phytochemicals and nutrients like the alkaloid, phenol, flavonoid, and antioxidant, analgesic, antipyretic, the antibacterial, anti-tumor, anti-arthritic, anti-diabetic, anti-emetic, the anxiolytic, and nephroprotective activity, anti-HIV, anti-eosinophilic, etc. And in a conclusion, friends, so we put the speculated that the Sosaria ballata may be a good natured source of the pharmacologically active the constituents, and it may be considered for the development of the herbal formulation against the various diseases. Thank you, sir. Thank you for giving me this opportunity to present here.
Good evening, everyone. I would like to thank organizing team of MIST 2020 for giving me this opportunity. Topic of my study is in silico validation of the efficacy of phytochemicals from Passiflora edulis SIMS against severe acute respiratory syndrome coronavirus 2. COVID-19 or severe acute respiratory syndrome coronavirus 2 represent highly contagious and deadly disease rapidly spreading affecting 216 countries. Origin from Wuhan city, China to rest of the world. COVID-19 is a positive sense single-stranded RNA virus enveloped with 60 to 140 nanometer in diameter. On basis of literature survey, Passiflora edulis SIMS selected for present study. This plant includes several properties including anti-inflammation, anti-cancer, anti-diabetic, anti-hypertension, and anti-aging. Hence, present study focused on finding phytochemical from Passiflora edulis SIMS against COVID-19. In vitro screening coupled with in silico screening is best option to validate drug activity of herbal medicine and identification of a lead molecule for further experiment leading to the drug discovery. There is only one way the world can exit this pandemic, and this is through scientific research. Identification of a potential target from COVID-19, retrieve phytochemical from Passiflora edulis SIMS, identify the hit molecules, analyze various properties of hit molecule, hydrogen bonding, molecular property, bioactivity, drug likeliness, ADME, find the best lead molecule against the target protein. 206 phytochemical information from Passiflora edulis were available in a various literature. 130 structure information about phytochemicals available in a database, PubChem. Main protease protein from severe acute respiratory syndrome coronavirus 2 was selected, as main protease play important role in viral replication, gene expression, and attractive target for severe acute respiratory syndrome COVID-2 drug designing. Three-dimension structure of a target protein, main protease obtained from RCSB PDB. Total 130 molecule from Passiflora edulis SIMS screen against target protein severe acute respiratory syndrome coronavirus 2 main protease protein via virtual screening. After docking process, molecule that showed free energy of binding greater than 6 kilocalorie molecule were considered as heat molecule. Top rank 5 molecule that possesses free energy of binding greater than 8 kilocalories per molecule selected for the further analysis. Analysis of 5 mark molecule from Passiflora edulis SIMS revealed that all top rank except phytofluin exhibited hydrogen bond interaction with target severe acute respiratory syndrome coronavirus 2. Main protease target protein phytofluin interact with 15 hydrophobic interaction with amino acid residue. Tannic acid exhibit 5 hydrogen bond and 11 hydrophobic interaction, whereas in vanillic 1 hydrogen bond was formed with 6 hydrophobic interaction. Longifluoroside A hydrogen bond 3 and 12 hydrophobic residue. Fluorescin showed 8 hydrogen bond and 5 hydrophobic residue. Dog complex of fluorescin with its target and ligand interaction diagram using ligplot tool. Dog complex of phytofluin with its target main protease and ligand interaction diagram. Dog complex of tannic acid with its target MPRO and ligand interaction diagram. Dog complex of vanillic acid with its target MPRO and ligand interaction diagram. Complex of longifluoroside A with its target MPRO and ligand interaction diagram. Among 5 molecules, 2 compound, prurithin and vanillic acid, totally passed all Lipinski rule and thus no violation. Tannic acid showed 4 violation, phytofluin and longifluoroside A showed 2 violation each. Bioactivity score of 5 compound were studied for its activity against 6 important drug classes. Larger score indicate probability that particular molecule will be active. Analysis of 5 molecules performed considering water solubility, where pleuresin found to be very soluble in water, phytofluorine and tannic acid insoluble, vanillic acid and longofluorescein A moderately soluble. In order to analyze drug likeness property, a set of rules were studied for these molecules and revealed that pleuresin and vanillic acid possesses best pharmacological activity as well as drug likeness property. Pleuresin exhibit 9 kilocalories per molecule free energy against target protein with 8 hydrogen bond and 5 amino acid involved in hydrophobic interaction. From 5 compound, 2 compound totally passed Lipinski rule and no violation. Further analysis revealed that pleuresin exhibit best drug likeness compared to all other molecules studied. Analysis against 6 drug target class revealed that vanillic acid and tannic acid were inactive against all drugs, whereas pleuresin, phytofluorine, and longofluorescein A found to be inactive against some target only. From top lead, only pleuresin was found to be very soluble in water. Order of the top lead can be pleuresin, longifluorescein A, and phytofluorine. Considering all criteria selected for study, pleuresin selected as the best lead molecule from the plant through the study. Thus, pleuresin can be considered further in vivo or in vitro studies against severe acute respiratory syndrome coronavirus 2 to confirm result of in silico and thus can be validated experimentally. References. Thank you.
Good afternoon, everyone. My topic name is Role of Oxidative Stress and Genetic Instabilities in Female Infertile Subjects and its Risk for Cardiovascular Disease. Background of the study. Infertility is a global health issue which is affecting approximately 8 to 15% of couples, and it is a multifactorial disease which is defined as the failure to achieve a clinical pregnancy after 12 months or more of unprotected regular sexual intercourse. Nowadays, cardiovascular disease become the leading cause of mortality, and it is reported that by the year of 2030, more than 22.2 million people will die annually from CVD. Growing evidence suggests that increased cardiovascular risk is associated with female infertility caused by conditions such as polycystic ovarian disease, obesity, thyroid dysfunction, and endometriosis. Moreover, the role of oxidative stress and somatic DNA damage in female infertility has not been studied extensively. Oxidative stress also considered to be an important risk factor for CVD. Hence, the present study was undertaken to evaluate the role of oxidative stress and genetic instabilities in female infertility and its risk for CVD. Hypothesis. In several studies, it was reported that infertility may share some common pathways with CVD. PCOS, obesity, thyroid dysfunction are associated with CVD. Oxidative stress play an important role in the development of CVD, and it is also associated with infertile patients with conditions such as endometriosis, PCOS, obesity, and unexplained infertility. In the present study, it was demonstrated that there is a significant increase in mean CBMN frequency and MDA concentration among study subject when compared to the control subject. Moreover, it was also observed that infertile study subjects with various risk factors such as age, birth order, family history of infertility, PCOS, endometriosis, history of diabetes, hypertension, dyslipidemia, etc. can lead to increased genetic instabilities and severity of infertility. Objectives of the study. CBMN assay is carried out to quantify the extent of somatic DNA damages among subjects with female infertility. Total cholesterol, fasting blood sugar is checked among the female infertile subject to assess the biochemical characteristics associated with infertility and its risk for cardiovascular disease. MDA assay is carried out to measure the extent of oxidative stress among infertile females and its risk associated with cardiovascular diseases. Materials and methods. In the present study, 45 infertile females are selected as study subjects and 30 healthy females are selected as controls. Various demographic, physiological, clinical, and lifestyle characteristics were recorded using Performa. After 8 to 12 hours of fasting, 7 ml of venous blood were collected And using this venous blood DNA damage study is done by CBMN assay, and MDA concentration was tested using MDA assay, and biochemical tests were conducted, which include fasting blood sugar and total cholesterol. Results. T-test was conducted between study and control subject, and it was statistically significant. The mean value of total cholesterol, MDA, mean CBMN frequency, and FPS showed a statistically significant difference among study and control subject. An elevated mean CBMN frequency was observed among subject with advanced age and subject with birth order greater than 4. And it was also observed that the people who reside in urban areas showed an increased mean CBMN frequency comparing to the rest. Study subject with sedentary type of occupation showed increased mean CBMN frequency when comparing to the rest with non-sedentary type of occupation. And study subject who attained menarche after 13 years showed elevated mean CBMN frequency. And it was also observed that subject with irregular menstrual period, subject with PCOS and endometriosis, the obese subject, and subject with which follow non-vegetarian diet have shown higher mean CBMN frequency when comparing to the rest. Subject with regular exercise and good physical activity showed decreased mean CBMN frequency, and an increased mean CBMN frequency was observed among study subject with mental stress, and there is an increased mean CBMN frequency with the history of diabetes, history of hypertension and dyslipidemia. And an elevated mean CBMN frequency was observed among subject with high concentration of MDA, higher total cholesterol and higher FBS. Comparison of quantitative variables between study and control subjects were analyzed using independent sample t-test. The results are expressed in mean and standard deviation. The mean values of FBS, total cholesterol, MDA, and mean CBMN frequency were showed a statistically significant difference among study and control groups. These are the graphs which shows distribution of mean CBMN frequency according to age, birth order, residence, and menstrual period. And these are the graphs which show distribution of mean CBMN frequency according to PCOS, obesity, and mental stress. And we can see there is an elevated level of mean CBMN frequency in subject with PCOS, obesity, and mental stress. And these are the graph which shows the high mean CBMN frequency of subject with a high level of FBS, total cholesterol, and MDA concentration. In the first image, we can see cytokinesis blocked by nucleated cells, and in the second cell, we can see micronuclei in it. Discussion. Several studies have found that age and obesity play an important role in infertility and CVD. Musa and Osman in 2020 found that age greater than 35 years significantly increased the risk for infertility, and they also observed that obesity play an important role in infertility. Dringa and Vasan in 2012 reported that increasing age is an independent risk factor for CVD. In the present study, it was observed that CBMN frequency was increasing with advanced age, and subjects who are obese have a high mean CBMN frequency. It shows that the infertile subject who's selected in this study have high risk of CVD in the future. In the study, it was observed that subject with family history of diabetes and subject with a high amount of FBS, total cholesterol, and high concentration of MDA showed elevated mean CBMN frequency, and their future risk for CVD is high. And this result was supported by several studies by Enslin et al. 2012, Gu et al. 1999, etc. Conclusions. Several factors that lead to infertility such as age at marriage, number of abortions, alcohol consumption, underlying disease and BMI. When comparing to fertile women, infertile women have a higher level of cardiometabolic risk factors. Lifestyle factors, demographic parameters and physiological characteristics discussed in this current study have variable effect on female infertility. Oxidative stress and DNA damage also showed an important role in female infertility. Lifestyle factors such as obesity, smoking, diet, alcohol consumption have been observed as infertility modifiers. Healthy lifestyle factors including exercise are associated significantly with reduced DNA damage. Identifying and controlling chronic diseases such as diabetes, hypothyroidism, hyperthyroidism increases fertility prospects. Reference. Thank you.
Hello everyone. So the topic of my presentation is virtual screening of natural compounds against Mycobacterium tuberculosis. So as we know, tuberculosis is an airborne infectious disease which is caused by Mycobacterium tuberculosis, which was responsible for around 1.5 million documented deaths in 2018. So as we know, India is one of the high TB burden countries, contributing to about 27% of the global TB patients. And the current situation, it calls for an urgent need to develop new anti-tubercular drugs with less side effects and shorter treatment duration. So the aim of the present study was to use in silico techniques for virtual screening of natural compounds obtained from Euphorbia millet and Plectranthus amboinicus for the discovery of potential inhibitors to enoyl ACP reductase, which is an enzyme involved in the biosynthesis of mycolic acid. So as we know, in silico techniques is based on theoretical interpretation and is performed in a virtual environment, and it can be used in all steps of drug discovery process up to preclinical testing, thereby reducing cost and time associated with the process. So to validate, it is to validate the drug activity of the medicinal plants and to identify the lead molecules. To develop the drug molecule, the best method is an in silico screening method. So the objectives are selection of the target protein and plant, retrieval of the phytochemicals compounds of Euphorbia millet and Plectranthus amboinicus from the databases, and preparation of the target protein and ligands for the docking process. Later we did the molecular docking with the target protein and ligand by using autodock Vina using the Pyrex interface, and later the visualization of the protein ligand interaction by using PyMol software, and its interpretation was done. So later we compared the hit molecules with the synthetic inhibitor molecule which was already present in the database. So this is the materials and methods. These are the main three steps. The first one is the selection and preparation of the target protein. Second is the preparation of the ligand molecule, and third is the molecular docking. So this is how we did. First we downloaded the PDB structure of the target protein. Second we did the primary and secondary structure analysis, and later we identified the active site, and then we went ahead with the preparation of the target protein using the autodock software. So the next step is the preparation of the ligand molecule. Here identification of the phytochemicals was done through extensive literature search, and retrieval of the details of the selected phytomolecules was done from PubChem, and retrieval of the structure of the compound using SMILES notation was also done. So last we checked the physiochemical properties in Swiss ADME. So this is how molecular docking was done. First we loaded the protein and the ligand in the Pyrex interface for the docking process, and the protein and the ligand was prepared, and once the docking process was started, we selected the active site residues of the target protein and initiated the autodock Vina. So once the analysis was done, we saved the protein ligand interaction in the PDB format, and later it was visualized using the PyMol software, and the hydrogen bond interactions was also obtained through the LickPlot Plus tool. So the results is a total of 106 phytochemicals from the two selected plants were used for in silico screening, out of which 85 phytochemicals were obtained from Plectranthus amboinicus and 21 phytochemicals were obtained from Euphorbia millet. All the screened phytochemicals, they showed a good binding affinity against the target protein, that is the enoyl ACP reductase, which was more than minus 6 kcal per mole, which justifies the traditional use of plant Euphorbia millet and Plectranthus amboinicus against TB. The inhibitor molecule 3KX was docked with 4R9R using the Pyrex interface, which also gave a good score of minus kilocal per mole. So out of the 106 compounds docked, the 3 compounds showed free energy of binding similar to and greater than the synthetic inhibitor 3KX, which was for 3KX it was minus kilocal per mole, except epigenin. So epigenin showed minus 8.1, whereas beta cytosterol and eufol, they showed greater binding affinity when compared to the synthetic inhibitor 3KX. So this is the diagram here. As you can see, this is the target protein, and this is the ligand which is bound to the target protein in the active site, along with the NAD molecule in the magenta color is present, so which is a cofactor, and this is the PDB structure of the target protein. It has 4 similar chains, out of which only one chain was taken for the study. So similarly, we have for UFOL, UFOL is bound to the active site, and this is the Lick plot plus analysis, where the green dots represent the hydrogen bond, and these represents the hydrophobic interactions of the protein. So this is similar for beta cytosterol. As you can see, this is the hydrogen bond between the ligand and the NAD molecule, and these are the hydrophobic interactions. So epigenin didn't have any hydrogen interaction, but it had all hydrophobic interactions with the target protein. So the conformation of the ligands with the target molecule showed the similarity of the pose with the active site when compared with the inhibitor molecule 3kx. So further validations are necessary, that is in vitro and in vivo studies, to propose these molecules as candidate drugs against TB. So the structure selected for the study was to which another inhibitor 3kx is attached, and this ligand 3kx is a direct inhibitor of the tuberculosis NITD 564. And also the beta cytosterol is a phytosterol with antimicrobial, anti-cancer and immunomodulatory, anti-diabetic, anti-cancer effect, and it is also used as an adjuvant to the treatment of tuberculosis. So hence from the present study, it can be concluded that UFOL and beta cytosterol can act as potential drug candidates against tuberculosis. The study clearly indicates that when compared to NITD 564, UFOL and cytosterol is showing similar activity and can be considered for further analysis. Synergistic activity of these two compounds should also be evaluated, and in order to substantiate the in silico results, in vitro and in vivo investigations are necessary. These are the references. Thank you.
Good morning, everyone. So my topic is the effect of aluminium and cobalt on the structural and magnetic properties of off-stoichiometric YKT Fe17 compounds. Why is it off-stoichiometric? Because these YKT Fe17 compounds are the famous 217. 2 stands for R2 rare earth and Fe17 compounds. But I have made Fe18 compounds. Let's go. So these 217 intermetallics are nothing but R2Fe17, which come under the class of RTM intermetallics, where R stands for rare earth and TM stands for transition metal. So these RTM intermetallics, they form in several specific compositions with a variety of magnetic structures. So for example, RFE2, which form in cubic, R6Fe23, which form in cubic structures, and this RCO5, R2Fe14b, R2Fe17, R3Fe29, R3Fe12, these elements, they come under the class of permanent magnet materials. So these RTM compounds, they exhibit interesting magnetic properties because of the combined effect of rare earth, which has 4 localized moments, and the transition metal, which has 3D itinerant moments. The interesting properties are the rare earths have large magnetic moments due to the localized 4f electrons and a large uniaxial anisotropy due to the strong crystalline electric fields, but they have low Curie temperatures because, as we have already discussed, the 4f electrons are highly localized, so the exchange is indirect or weak exchange, because of which they have low Curie temperatures. Whereas the transition metals, they have low magnetic moments due to itinerant of free 3D electrons and low anisotropy due to the quenching of orbital angular momentum, but they have high Curie temperatures because of the strong and direct exchange. So as we have discussed, these 217 compounds, they are useful for permanent magnet applications, and the basic requirements for permanent magnetic applications are they should possess large saturation magnetization, high Curie temperature, and large uniaxial magnetocrystalline anisotropy. So among these 217 intermetallics, they have high saturation magnetization compared to all the other RFE intermetallics, but they have low Curie temperature and small and planar anisotropy. So to improve these properties and them as probable candidates for permanent applications, we can improve by choosing a proper rare earth and by substituting magnetic elements such as cobalt and nickel for iron, and also by substituting a non-magnetic element such as aluminum, gallium and silicon for iron, and thereby improving the TC and decreasing the planar anisotropy and thereby increasing the uniaxial anisotropy. And also by interstitial modification by nitrogen and carbon also we can increase the Curie temperature. So these all are reported. And what I have done is, instead of substitution of this transition metal, I have added the aluminum. Instead of making R2Fe17, I have added R2Fe8 compounds. In general, the structure of 217 compounds depends upon the type of rare earth that we use. If the rare earth is heavier than Tb, they form in hexagonal structure, and if the rare earth is lighter than gadolinium, they form in rhombohedral structures. Structural studies. So by employing XRD, we understood that all the compounds are formed in hexagonal structure, and we can see that the peaks are slightly shifted towards right side, which indicates that with the substitution of cobalt for iron, the size of the unit cell has reduced slightly because the cobalt size is slightly, and the extra site was perfectly suiting for aluminum, and you can see there is no much. It is exactly fitting the XRD. So these are the lattice parameters of the rate well defined this YGTFE17A. And here we can see that by adding extra aluminum, the unit cell volume has increased from 612 to 638, whereas by substituting cobalt, there is slight decrease in the unit cell parameters, which is due to the smaller size of cobalt compared to iron. Coming to magnetization studies, the saturation magnetization seems to be increased with the addition of aluminum as well as with the substitution of cobalt to these compounds. The increased magnetization with the addition of aluminum can be attributed to magnetovolume effect. We understood just now, we have seen that with the addition of aluminum, the cell parameters have increased. So the increased saturation magnetization can be attributed to magnetovolume effect, whereas the increased magnetization with the substitution of cobalt could be due to the additional exchange, that is FeCO and CO exchange in addition to FeFe already existing exchange. Okay, even though the same thing could be observed in Curie temperature, with the addition of aluminum there was a slight decrease in the Curie temperature, but with the substitution of cobalt there is an increase in Curie temperature from 324 to 705 Kelvin, which could be attributed to the higher exchange, that is CO and FeCO exchange in addition to the iron exchange. Okay, coming to anisotropy studies. The anisotropy studies were done on the magnetically aligned samples. Okay, when the sample powders were mixed in the epoxy resin and the grains were aligned in the presence of external magnetic field by using a BH loop transistor up to a magnetic field of 5 Tesla. The aligned samples, when the field is applied perpendicular to the grains, is used to know the easy magnetization direction. And when the magnetic field is applied parallel to the direction of these grains, then that is used to understand the anisotropy fields. So from the XRD studies of aligned compounds, we understood that all the samples have a planar anisotropy, and the anisotropy fields can be measured by measuring the can be understood by measuring the area between the H parallel and H perpendicular. Here we can say that with the substitution of cobalt, there is a slight increase in the area between H parallel and H perpendicular, which indicates that the anisotropy increased with the substitution of cobalt, which is a very slight increase. This could be attributed to the cobalt. The structure of cobalt is hexagonal, whereas iron structure is cubic. So this could be due to the structural change. Okay, thank you.
Good morning, respected sir and madam. I am Aritropal from the School of Pharmacy Department. of the Newton University. Today I am going to represent the medicinal value of Centella asiatica. I worked with Mr. Parag Ghosh and Dr. Subhashish Mondal in the presentation of this poster. At first, I want to say that why I choose the herbs for my poster. As we know from the very ancient time, human beings are dependent on plants in every prospect of life. They provide us oxygen as well as medicines. They are the purest source of medicinal constituents, so scientists started their research work on plant based drug discoveries as they are non synthetic, less toxic, and with almost zero side effects. Now I want to come to my topic, which is Centella asiatica. It is a well-known herb of Apiaceae family which can be found mainly in Asian countries like India, Sri Lanka, Indonesia, etc. It's commonly known as gotukola or thankuni. As we can see, it has small fan-shaped leaves and pink or white flowers. It belongs to the plantae kingdom. It is a angiosperm of Magnoliopsida class having the genus Centella. It has almost 20 different species. The main motto of this presentation is to overview the pharmacological activities and the medicinal values of this plant. And for this presentation, we took the help of Google Scholar, Science Direct Database, Wikipedia, and some more sites like this. As per the studies, we came to know that this small plant is a storehouse of several phytochemicals like acetic acid, medicacic acid, aceticoside, thankunic acid, isothankunic acid, and much more. Due to these phytochemicals, it shows antioxidant, anti-diabetic, anti-inflammatory, wound healing, anti-ulcer, immunostimulant, and many more properties. It also shows anti-cancerous and neuroprotective activities and also can be used in treatment of skin disease. According to Rahman MM et al., anti-diabetic properties of leaves extract of Centella asiatica was evaluated in Alexon-induced rat by administering extract at a concentration of 250, 500 and 1000 mg per kg. After 3 hours of ingestion, reduction in blood glucose level was noticed by 32.6%, 38.8% and 29.9% respectively. From another et al. By Orhan IE, we came to know neuroprotection aspect of Centella asiatica mainly involves enzyme inhibition, prevention of amyloid plaque formation in Alzheimer's disease, dopamine neurotoxicity in Parkinson's disease, and reducing oxidative stress. Here, the graphical representation of different pharmacological activities of Centella asiatica is shown. At last, we can conclude that the immense medicinal potential can't be judged by the small size of Centella asiatica. Due to the presence of wide bioactive compounds, the plant have vast application. The plant can be a safer alternative for the formulation of new drugs. Further research is needed to confirm their activities mentioned in ancient scripts, followed by clinical studies for their safe application. The plant should be preserved in every possible way. Thank you.
Very good afternoon to everybody. A research scholar from Ranchi University, Jharkhand. I am here to present my paper on the topic Study on Importance of Aquatic Beetles in Freshwater Ecosystem.
Background of the study. Freshwater ecosystems perform a number of environmental functions. These are colonized by a diverse group of aquatic insects which spend their whole or some part of life cycle in or on these water bodies. Among these, the aquatic coleopteran are the most colorful and interesting group. Important aquatic families are Dithycidae, commonly known as diving beetles, Hydrophilidae, commonly known as water scavenger beetles, Haliplidae, which is also known as crawling water beetles, Gyrinidae or wriggling beetles, Scirtidae, known as Mars beetles, and Alimidae, which are also known as reaper beetles. They are of great importance as they serve as food for fish and other invertebrates. They are very good indicator of water quality and pollution. Not only this, they also help in processing and cycling of nutrients in the water ecosystem. In spite of these contributions, very little attention has been paid on them. Thus, this study will provide a fresh outlook on their importance in the freshwater ecosystem. Going to the hypothesis. Insects are the most diverse group of organisms in freshwater bodies. They are the best indicator of the biotic environment of the water body. They play an important role in ecosystem functioning, for example, nutrient cycling, primary production, decomposition, and material translocation. Present study will highlight on these facts with special reference to aquatic beetles. Coming to the objective. This study will investigate the importance of aquatic beetles as bioindicators, biomonitors, predators, and biocontrol agents. Aquatic beetles as bioindicators and biomonitors. Biomonitoring or bioindication is the science of applied ecology using organisms that live within natural ecosystem to monitor any change in the ecosystem. Monitoring water quality by conventional chemical methods can tell us very little about the effects of pollution on aquatic life. Aquatic insects have been used effectively as biological indicators to determine environmental condition of water bodies. These are a diverse group and excellent indicators of habitat quality. These fulfill the required characteristics of bioindication, such as species richness and diversity, easy handling, fragility to small environmental changes, and good organism responses. Using beetles as indicator organisms is very beneficial because they have limited mobility or are sedentary, and therefore they can become good assessors of pollution impact on a specific habitat. Using them as bioindicator is environmental friendly also, and this is less expensive. This is why they are generating great interest in researches regarding conservation of aquatic ecosystems. Role of aquatic beetles in food chain. These beetles form an important component of the aquatic food chain and energy pathways. They comprise a high proportion of biomass in freshwater ecosystems. They belong to different feeding groups such as shredders, filter feeders, deposit collectors, and predators. During larval stage, they constitute the main nutritive fauna of fish and play a significant role in the processing and cycling of nutrients. They process dead and decaying woods and leaf litters. Nutrients processed by them are further degraded into absorbable form by fungal and bacterial actions for use by aquatic plants. Aquatic beetles are also a very good biocontrol agent. Aquatic insects have been introduced worldwide to control the noxious waves which create great problem for the native species and water bodies. Use of herbivore species of insects have shown good results. Further studies are going on to prove the usefulness of aquatic beetles in this field. Coming to the conclusion of the study. The study provides a baseline about the importance of aquatic beetles in the freshwater ecosystem. It concluded that these are excellent indicators of water quality and perform valuable functions for the maintenance of the aquatic ecosystem. In spite of this, number of researchers working on beetles is very low, making it difficult to achieve adequate knowledge on this group. Basic knowledge towards them is necessary as they can guide the formulation of conservation measures. In view of this, there is need to encourage more work on beetles. Thank you.
Good afternoon, one and all present here. First of all, I would like to thank you for giving me an opportunity to present my ideas and views. Let me introduce myself. I'm Soumya P. from Kerala. I'm doing my second year MSc in Bioinformatics at Sri Krishna Arts and Science College. Today, I'm here to present a paper on computational analysis of molecular discovery to treat rheumatoid arthritis and initiation towards rational drug design. I would like to give a brief introduction. An autoimmune disease is a condition in which your immune system attacks your body by a mistake. The immune system normally guards our body against germs like bacteria and viruses, but in the case of autoimmune disease, it recognizes our own healthy cells as foreign invaders and attacks them by mistake by releasing autoantibodies. There are about 80 types of autoimmune diseases present, including rheumatoid arthritis, lupus, etc. Rheumatoid arthritis is a long-term, progressive, and disabling autoimmune disease. It causes inflammation, swelling, and pain in the joints of our body, especially in the hands and feet first. There are mainly two types of RA present. RA is estimated to be affect approximately 0.24 to 1% of the population and is more common in women compared to men. The risk factors includes genetic factors and hormones, and it varies from lifestyle habits such as stress and obesity to the gut bacteria present in your body. The symptoms are characterized by tenderness, pain, swelling, redness, etc. in our joints, slight fever, weight loss, etc. The diagnostic methods includes diagnosis of high level of C-reactive protein in our body, MRI scan, where bone edema or erosion is visible, etc. And the treatment is of two types as monotherapy and combination therapy. In monotherapy, disease-modifying anti-rheumatic drugs, which slows down the progression of the disease in your body is used, such as hydroxychloroquine or leflunomide. And in combination therapy, a combination of both the DMARD and a biology therapy is used. There are a couple of biomarkers present in our body. These are associated with rheumatoid arthritis. Biomarkers are molecules which can be used as an indicator of severity or the presence of some disease state in a patient's body. The biomarkers associated with rheumatoid arthritis includes rheumatoid factor, cyclic citrullinated proteins, antibodies, 1433 ETA protein, etc. And these are all the gene and miRNA biomarkers associated with RA. And protein biomarkers are more popular because of the availability of a large range of analytical instrumentation. The protein biomarkers includes resistin, SAA, calreticulin, galsolin, ACPA, etc. And for many of them, crystal structures are available in the online databases, and these can be retrieved at any time. And they perform a lot of important function associated with the rheumatoid arthritis. And to explain marine peptides, they are important bioactive natural products which are present in many marine species. They have high potential nutraceutical and medicinal values because of their broad spectra of bioactivities. And we can use both immunomodulant and immunosuppressant drugs. An immunomodulant is a substance that affects the function of the immune system, whereas immunosuppressant drugs are a class of drugs that suppress or reduce the strength of body's immune system. The objective of this study is to know about the biomolecular interaction of natural peptides with the protein biomarkers associated with the rheumatoid arthritis. For this, we performed molecular docking. Molecular docking is a kind of bioinformatic modeling which involves the interaction of two or more molecules to give the stable effect. Depending upon the binding properties of ligand and target, it predicts the three-dimensional structure of any complex. And protein peptide docking is where we perform the molecular docking of a protein biomarker and a peptide, marine peptide string. We use tools such as Capstock and LigPlot for the docking purpose. Capstock is a web server which provides an interface for modeling protein peptide interactions. And LigPlot is a computer program that generates schematic 2D representations of protein ligand complexes. And we perform this docking in different combinations of protein targets and peptide sequences, and we have obtained results in both LigPlot and Capstock. And now coming to the discussion, we used 7 protein targets and 10 marine peptides, and we have obtained 31 interacted peptides, and all these 31 interacted peptides have hydrogen bond interactions present in them. So to conclude, among the various interactions of target and peptide, only a number of peptides shows a better interaction, and those peptides can be taken for further studies in vitro and in vivo. They can be used as a potential drug candidate in the future for the rheumatoid arthritic disease. And the marine compounds also have the potential to be used as a drug candidate for various disease in future. Thank you.
Good afternoon, everyone. I am Shashi R, Research Scholar, Department of Chemistry, Bangalore University. I would like to talk on my research topic, synthesis and crystal structure of 2-acetyl 6-ethoxycarbonyl 5-2-fluorophenyl 3,7-dimethyl 5H thiazolopyrimidinium chloride. I am working under the guidance of Dr. Noor Shaina Begum, Department of Chemistry, Bangalore University. My outline of my talk is introduction, synthesis, spectral analysis, crystal structure analysis, conclusion followed by references. To begin with, let's see what is the uses or the applications of thiazolopyrimidine compounds. So to begin with, thiazolo group is an heterocyclic compound which contains 2 heteroatoms such as nitrogen and sulfur atom, which is a part of 5-membered aromatic ring. And pyrimidine is also an heterocyclic compound which is having 2 nitrogen atoms as the heteroatoms, and both these compounds exhibit a wide range of biological activity. Therefore, we decided to combine both pyrimidine as well as thiazolo, which results in thiazolopyrimidine moiety. This thiazolopyrimidine moiety is also having great biological activities such as anti-inflammatory, antihypertensive, antimicrobial, anti-tumor, and it also exhibits properties such as calcium antagonist, and it can also act as HIV reverse transcriptase inhibitors, and some of the thiazolopyrimidine derivatives have been reported as acetylcholine esterase inhibitors. So to begin with the synthesis, we have synthesized different types of dicarbonyl derivatives of thiazolopyrimidines, and the starting material being the 3,4-dihydropyrimidine, which is one of the starting material for the synthesis of thiazolopyrimidine compounds. This 3,4-dihydropyrimidine thiones were synthesized by one of the well-known reaction which is known as Beginnery reaction. This reaction is a multi-component, one-part acid-catalyzed reaction where there is condensation between an aromatic aldehyde, urea or thiourea, along with an active metallic group with a dicarbonyl group. This reaction is catalyzed by concentrated HCl or any other strong acids. So once dihydropyrimidines have been synthesized, we fuse these DHPMs with dicarbonyl group or dicarbonyl compounds such as 3-chloro-2,4-pentadione in triethanol for 6 hours, which results in formation of thiazolopyrimidine compound, which is compound 3. We have synthesized 10 compounds of these kind, and these compounds are biosteric analogues of purines and are potentially bioactive molecules. The chemistry of thiazolopyrimidines have received much attention because of its convenient ability through the most versatile synthetic method developed by. We have further proved the structures by spectral analysis such as IR, where we can see the functional group as ester at 1690 and a ketone stretching frequency at 1665, which shows the presence of the compound. And we further proved it by NMR analysis, where we have the aromatic region between 7 to 8. We have the chiral hydrogen between 6 to 7, and we also see the methyl protons and CH2 protons between the region 2 and 3, which has been expanded here. And we also have the dimethyl group, which is between 1 to 2. Once these compounds were being spectrally analyzed, we further carried out another technique, which is single crystal X-ray diffraction technique, where we could exactly tell the structure of the molecules more precisely. So this X-ray, once the crystals were obtained, X-rays which were placed between the LED screen and the photographic plate were bombarded with X-rays, and once the X-rays came in contact with the crystal, it gave rise to diffracted X-rays patterns on the photographic plate. These photographic plates, which we usually call it as electron density map, was further processed to give the exact structure of the molecule, which is known as structure, solution, and refinement. So this is the schematic representation as to how we obtain the exact structure of the molecule using single crystal X-ray diffraction. So there are various softwares which are used in order to solve a structure by single crystal X-ray diffraction. Initially, data collection is done by SMART, data reduction is done by SAID+, data absorption correction is done by SEDAPS, structure solution is done by SHELX S97, structure refinement by SHELX L97, and graphics such as the ORTEP diagram of molecule, and packing diagrams, which can be explained using diamond and mercury. The mean plane calculation was done by PAST. SIF is generated by NFR. The information obtained by single crystal structure analysis are unit cell dimension, crystal density and chemical composition, crystal symmetry space group, fractional coordinates, molecular geometry, bond angles and bond lengths, stereochemistry, intra and intermolecular interactions, polymorphs, chirality, and absolute configuration, along with packing and disorders in the crystalline structure also can be obtained. And this is one of the most precise method or a technique which is used to determine any kind of structure such as organic molecules. So our representative molecule was subjected to single crystal X-ray diffraction, where we obtained a crystal system of triclinic, space group of P bar 1, a dihedral angle between the pyrimidine ring and the aryl ring to be 89.85 degree, and the pyrimidine ring had a boat conformation with N2 and C5 deviated from their mean plane. Since there were a number of heteroatoms such as nitrogen, sulfur, fluorine, and chlorine, we expected a number of interactions, where we were also satisfied to see three different interactions such as CHF, CHO, and NHCl. And as you can see on the screen, we have a trifurcated bond which is being shown here, and O2 prime is acting as the acceptor for all the three carbons, all the three different carbons such as C13, C5, and C19. And the R factor was found to be 7. Conclusion: we have attempted the synthesis of dicarbonyl derivatives of diazolopyrimidines. The compounds synthesized were obtained in good yields. In addition to spectral technique, we also were successful in order to get the single crystal X-ray diffraction technique done for the few of the compounds which confirm the presence of proposed ring system. And these are the references and some of the papers published by our lab mates on the same topic, thiazolopyrimidines.
Thank you all. Due to time constraints, we are postponing this oral presentations number 64 to 75 to the next session. So please stay tuned for the last session